Hello, hello friends. Myself Dr. Vinayaka. In the previous classes of the plant physiology, we are studied the different aspects of the physiology, just like transportation and also transpiration, ascent of sap, guttation, plant water relationship, and the plant mineral nutrient. So this is the continuation of that part, that is the plant enzymes. This is an concept of the plant physiology, in that you will study the different types of the enzyme and their enzyme activity and the mechanism of action. This is the syllabus of the physiology for this semester, that is plant enzymes, the properties of enzyme, nomenclature, structure, composition of enzymes, coenzymes, cofactors, and mode of action, enzyme inhibitor, and factor affecting on the enzyme activity, that you will study in this chapter. So going to the enzymes, so first, so what are enzymes? Simply, enzyme are defined as biocatalyst. Then you have a question, what is a catalyst or what is a biocatalyst? Catalyst is nothing but these are the substance that will be increase the activity. There is a reaction is showing that the methanol to formaldehyde by using the oxygen in the air. So here the oxygen is act as a catalyst. So what will that will increase the rate of reaction. So like that, these enzymes will act as a biocatalyst naturally, or we can induce the reaction by the enzymes, and that will also increase the activity. So going to the what is a biocatalyst? Biocatalyst is nothing but the capable of enhancing the rate of reaction without itself undergoing any changes. Enzyme will not changes, but the rate of reaction will be increases. So okay, going to the concept or the meaning of the enzyme. So enzyme is derived from an Greek word, and it means in living. En means in, zyme means living. The totally enzyme means in living. That means some of the substance which will be living inside the body or the inside the reaction. Okay, so going to the biocatalyst, so simply already told that these enzymes are the biocatalyst, and usually have the high molecular weight of the protein. There may be some exception, that may be ribosomes or RNA enzymes that may be have an exception for this quote. For the first time in 1860, Louis Pasteur has mentioned about the fermenting factors. He don't know exactly what will be happen, but he mentioned some of the factors that will be increase the activity of the reaction. So that he called as a fermenting factor. And later, the Kuhne in 1878 has coined the term enzyme. And the first time from the yeast cell in 1897, Buchner has extracted the enzymes. Buchner has extracted an enzyme that name called as zymase. From where he extracted? So he has extracted that enzyme from the yeast cell. And the first time these enzymes are purified by the James Summer in 1826, an enzyme called as urease, that was first crystallized enzyme, and James was crystallized this enzyme from the jock bean meal. Okay, jack bean, he is isolated from the jack meal and crystallized that enzyme. That name is, the enzyme name is urease. Okay, what is the enzyme name? Enzyme name is a urease. Okay, So there will be some specialized quotation for the enzymes. That is, all enzymes all enzymes are protein, but all proteins are not enzyme. The name itself that indicated that all enzymes, whatever the enzymes you will be study, all are protein, but all proteins are not enzymes. So this is an universal concept, but there may be exception. Earlier I told that some of the enzymes may be properly not the protein. There is an enzyme which is not a protein. That's first time was discovered by Altman in 1983. That's a ribulose P. That is not an exception for the enzyme. So going to the properties of the enzyme. So these enzymes are the protein with the catalytic power. Enzyme is nothing but the protein materials. They are colloidal in nature and they will have the high molecular weight. They will have the high molecular weight. And usually these substance on which they act is called as substrate. Okay, enzyme which they act, those are called as the substrate. So and usually biochemical reaction occurs in a bioactive sites. So usually in case of the enzyme, these activity occurs in a site called the enzyme. Sometimes there may be a specificity in this site. So going to those specific activity of the enzyme. So one is first is stereospecificity. Enzymes are very peculiar to the stereo nature. Already maybe some of the time you will study in the chemistry, that is stereochemistry. That stereochemistry indicated that the rotations, that may be L and D rotation. For that these enzyme activity will be specific. They have some specific activity. That's a stereospecificity. That means they act only one isomer, that may be L amino oxidase and the D amino oxidase. So like that there will be have some specific sites or the activity. And next one is reaction specificity. These are the some specialist or the specific substance. Same substrate can undergo different types of reaction using the different enzyme. That means similar type of the reaction will carry with the different enzymes. For example, glutamase dehydrogenase and aspartase glutamic acid glutamate amino transferase. So like that there will be some reaction specificity will be there. The next one is a substrate specificity. In that there may be a different types of substrate specificity. That's a one is absolute specificity. Here only one substrate will be specific to one reaction. That may be urease or maltase, so like that. And the relative specificity, here there will be a structure related to the substance will be specific. Example, trypsin. And generally the broad specificity, this is closely related substance, that's a hexokinase. So like that there will be a specific activity of the enzyme will be more. That's the stereo specificity, reaction specificity and the substrate specificity. So like that there may be amorphic molecules, enzymes are thermobable, and also solarization and crystallization of the enzymes may be occurs. So usually enzymes that will have the some specific unit. So usually the enzymes are referred in the different units. That's spectrometric units. That's a 1 UB used to Cuttles. That's a K8 is equal to 1 mole of substrate converted to the product in 1 second. So that is generally 1 IU is equal to 60 microcat. So like that there will be some specific enzyme unit will be there. And the enzyme numbers, that may be a turnover. Usually enzymatic reactions are reversible, indicate the catalytic power of the enzymes, index for the catalytic power, and carbonic anhydrous have the high turnover number. So based on the reactions or enzymes is differentiated into different types. That is, some of them are called as exoenzyme and some of them are called as endoenzymes. Exoenzymes is nothing but the enzymes secrete outside the cell. Okay, some of the enzymes they will secrete it outside the cell. So those are called as exoenzymes. That's the bacteria and yeast. They produce some of the enzymes. And endoenzymes, majority of the enzymes are endoenzymes. They secrete inside the cell. So usually, especially based on the first one, that's the types is based on the reaction. The second type is based on the chemical nature. So that's a induceful enzymes and also the constitutive enzymes. That's the induceful enzyme. The effect and the concentration of the enzymes depend on the concentration of the substrate. Example, nitrate reductases, and it is only inducible enzyme in the nature. And constructive enzymes, the effect and the concentration of the enzymes does not depend on the concentration of the substrate. Here what happens, the enzyme concentration does not depend on the substrate. You will have some concept just as zymogens. This is an enzyme. Some of the enzymes are found to be inactive in stage. Those enzymes are called as zymogens, or it is also called proenzymes. Usually, especially the digestive enzymes, that's a lipsin and trypsin. So those are referred as the zymogens. And zymogens and HCl helps in pepsinogen to convert pepsin. So like that, trypsin helps in a chymotrypsinogen to convert chymotrypsin. So there will be usually these are the enzymes, they involved in the conversion of the active form, inactive to the active form. So those enzymes are called as zymogens. And next, going to the order of reaction of the enzyme. That's the first order, second order, and the zeroth order of the reaction. So what is the first order reaction? The reaction which proceeds at the rate of proportional to the one of the reactant. That means in the first order reaction, usually the rate of proportional is equal to the one of the reactant. In the second order reaction, the which proceed at a rate of proportional to the concentration of the two of the reactant. The first one is only one of the reactant. Here there will be a concentration of the two reactant. Zero order reaction, the reaction which proceeds at a rate proportional to the either or any one of the reactants. Here they may be take one or maybe zero of the reactant. So that's how actually the order of reaction will be takes place. Next, signal of the substrate reaction. Usually the single substrate reactions, what happens, for example in case of sucrose, there will be a different mechanism will be occurs. That's usually converted into a in the enzyme called sucrase. Sucrose is converted into a glucose and the fructose. So like that there will be a changes in the product by substrate. Here there will be a two substrate. So based on the chemical constitution, again the lichens are enzymes are divided into different types. So that's simple enzymes, holoenzymes, apoenzymes. So like that there will be a different classification. The simple enzyme means made up of only one protein, that's a trypsin, lignin, etc. Conjugated enzymes means they constitute of proteins and the non-protein part. So that enzyme constituent protein and the non-protein part, those are called as a conjugated enzyme. Simple enzymes, there will be only one protein molecule, example trypsin. The holoenzyme, the holoenzyme is nothing but apoenzyme plus prosthetic group. Enzyme protein plus prosthetic group, that is called as a holoenzymes. Apoenzymes, so usually they will consist only protein part. In the holoenzyme, the apoenzyme consists of protein part, and another one is a prosthetic group. The mixture of these will be formed holoenzymes. Then you will have a doubt, what is a prosthetic group? Prosthetic is nothing but the cofactor or the coenzyme. The next one type is a dialysis. That's the separation of the macromolecule from the micromolecule. These are different types of the enzymes. So next is a enzyme as a metal activators. Enzyme requires some of the metal activator for their activity. That may be kinase required magnesium, zinc, manganese for their increasing the activities. Like that, aldose require zinc and catalase require the ferrous. Metallo enzymes. So some of the enzymes have the metal within their structure itself. So example, in case of Rubisco has the magnesium as a structure one. So those are called as metallo enzymes. Coenzymes. There is always a nucleotide present with the enzyme. So those part is called as a coenzyme. Exception for the iron. So usually how the vitamins, especially the vitamin B complex, have this type of coenzymes. So usually when the coenzyme derivatives plus substrate gives the coenzyme plus substrate plus B product. So like that will be occurs. So there are different types of the coenzyme, that may be NAD and NADP. So like that FAD, FMN, and also some ascorbic acid in case of vitamin C, and also the pyridoxal and biotin. So these are the some different types of coenzymes. So this is how actually the complete account on the enzyme types, enzyme definition, and the enzyme characteristic. So next, going to the enzymatic action. So in the next class we will discuss the enzymatic action.